Thank you very much for the introduction, and I'm very pleased to be here and honored to participate in the research and development programs of Gain Therapeutics. And I'm especially honored to follow the wonderful presentation that you've just heard. So my topic today is the role of allosteric drug discovery in targeting undruggable proteins in oncology. And what I'd like to do is focus on opportunities for the Gain Therapeutics platform of site-directed enzyme enhancement therapy, that platform and related proprietary technologies in the overall development of new therapeutic interactions. Next slide, please. So we've heard a little bit about the potential advantages of allosteric drugs versus traditional orthosteric drugs. And what I'd like to do is briefly recapitulate a definition of terms. So allosteric regulation refers to the modification of protein activity through the binding of a ligand, sometimes called an effector, to a site topographically distinct from the active site where molecules traditionally bind in either the setting of enzymes or certain cases receptors. The word allosteric, as you've heard, really derives from the Greek for other site and is used to emphasize this difference. The alteration of protein activity is typically accomplished by the reversible alteration of the protein conformation that accompanies effector binding. And you've heard some wonderful introductions along this line. There's a little bit of a schematic on the right hand, but you've already heard a number of the issues and how this works. Now, I'd like to give you my own analog, my own analogy of what allosteric inhibition really is. And all analogies are fraught with potential issues, but this is how I would like to at least put on the table how we can view allosteric inhibition. So imagine that you have an extraordinarily bright light bulb on the second floor of your house, and it's on in a way that you can't control. It has a switch which is difficult to get a hold of, and in any event, has been wired in such a way that when you try to turn that switch off, many other lights in the house go off. And what an allosteric intervention could represent is essentially finding a dimmer switch, perhaps in your basement, that can exquisitely control the obnoxious light in discussion. So in other words, you can modulate a function that you either wish to enhance or depress, but you can do so in a way that is at a distance from the functional portion of the target that you're discussing. Next slide, please. So there are potential advantages of the allosteric drugs that we are talking about in medical oncology, and they provide a very important new dimension. I, at this point, might remind the audience that medical oncology and oncology drug-related discoveries are an extraordinarily rapidly growing phase of entire pharmaceutical development. And there have been astonishing levels of public health and commercial advances in the field of medical oncology, and we expect that to continue. In fact, I could at this point briefly introduce the notion that President Biden has recently launched another program aimed at reducing the death rate from cancer by at least 50% in a reasonable period. And I personally believe that Gain and its platform can play a significant role in this goal. So let's go over some potential advantages of allosteric drugs in medical oncology. Some of this will recapitulate what you've already heard. There's greater specificity and selectivity of activity. Targeting allosteric sites may provide greater specificity and selectivity of activity, allowing for better efficacy and a better safety profile. Now, I want to emphasize that selectivity is a key advantage. A protein's active site can appear similar to other proteins in its family, making it difficult, sometimes in the extreme, to limit off-target and off-tumor effects. An allosteric site is more specific to an individual protein, potentially leading to safer and more efficacious medicines. An allosteric intervention may modulate a protein's activity in new ways. One modulator may shift a protein into a conformation that shuts it down entirely, while another, under the right conditions, may potentiate activity where such activity is desired, and any measure of activity in between. Another key feature is that one can identify previously inaccessible drug targets. So we've already heard the term undruggable. So allosteric drugs open the door to identifying drug targets that are inaccessible to traditional active inhibitors. And that's a point that we need to keep in mind. And also, I believe this offers a new foundation for combining oncolytic agents. So there's a potential to achieve important additive and synergistic effects and to prevent or overcome additional orthosteric anti-cancer agent resistance and so forth. So next slide, please. The need for novel kinase inhibitors is just one example of what may lie in store and the solutions that we may have. So the future success in kinase drug discoveries, which is a very important component of modern medical oncology, will depend on finding new ways to identify chemically more diverse specific inhibitors with good physical chemical properties, and I might add good pharmacokinetic properties. Finding compounds that are not already covered by intellectual property claims is also an important feature. And finding novel approaches to address resistance is also extremely important. One of the problems that we face in medical oncology and therapeutics related to cancer interventions is that when patients have advanced disease, to a very large extent, drug resistance can become an enormous problem. And it is one of the reasons why when patients have advanced tumors, especially visceral advanced tumors, often they will die of their tumor unless there's a comorbid event that takes them off first. So these are significant challenges that I believe allosteric approaches can benefit. Next slide, please. Allosteric modulators, continuing the theme for kinases, there are opportunities in non-ATP binding sites. Compounds which bind to sites distinct from the ATP site can interfere with catalytic or non-catalytic functions of the kinase, interfere indirectly with phosphorylation of substrates, and essentially be either inhibitors or activators, depending on what is required in a given situation. So allosteric modulators can lead to compounds that can be exquisitely selective, have different chemical structures to ATP site inhibitors, have different physical chemical properties, and can lead to a more specific functional readout and be helpful to overcome resistance, a key feature of the challenges we face. And because of their selectivity, become great tool compounds for target validation. Next slide, please. We can also discuss the notion that there is great potential for kinase agonists. Stimulation or enhanced regulation, potentiated regulation of a kinase pathway may be beneficial in some pathologies and could be essentially achieved by stabilization of the inactive site of the kinase, stabilization of the active state of the kinase, facilitating activation of the kinase. Activators will necessarily be compounds that bind outside the ATP site in our general discussion. And there could lead to the discovery of novel kinase activators, which in turn can provide insightful tool compounds and the identification of kinases whose activation can be therapeutic advantages that is hampered by currently by the lack of the effective tool compounds that we currently face. The discovery of novel kinase activators may therefore not only result in drug candidates, but could provide products, tool compounds to study the basic biology of kinase activation, leading to ever-increasing research opportunities. Next slide, please. So there are certain examples that we already have and that give us enormous optimism in understanding where target allosteric discoveries can lead. And the first, second, and third generation of drugs have in some ways failed to produce therapeutic benefits due to EGFR mutations. I don't want to, in this context, say that we're dealing with total failures. That's not what I'm saying. But we are dealing with very significant challenges. And I think all would agree that we need to approach this challenge in more effective ways. So targeting allosteric sites has gained attention for the therapeutic relief for many indications. The vast range of benefits targeting allosteric site essentially hints of the potential to serve as a strong target site for diseases with high mortality rates, for example, in non-small cell lung cancer. And these are important considerations in both therapy and advancing the public health. Next slide, please. There's novel target opportunities, novel target space within what we call the kinome. So this can be broken up into at least three portions. There are, in the setting of untargeted kinases, there are more than 500 kinases, but only a fraction of these have been targeted for drug discovery to date. So there's a significant potential to find new kinase targets. We can also begin the challenge of dealing with atypical kinases, that is, kinases with low sequence homology have not been well characterized, and due to the special features, it should be possible to design selective ATP site inhibitors for this class of kinases. Finally, there is the whole topic of pseudokinases. That applies to the notion that are key modulators in many signaling pathways through roles in scaffolding and in some cases, weak residual enzymatic activity can provide opportunities for targeting specific phenotypic readouts. Next slide, please. Allosteric modulators have the potential to combat drug resistance mutations. I've approached this several times because as a medical oncologist, I cannot list the number of patients whom I have treated personally, who despite even early responses have succumbed to a phenomenon of drug resistance, sometimes providing resistance against all known agents. And this, as a clinician, can be a very challenging phase of medical care. So what we can talk about is already some progress. For example, a gatekeeper mutation for the T31 to treat T314 mutation of BCR-ABL. This causes resistance to a set of clinically approved orthosteric drugs that are already on the market, the lead of which was imatinib, and these can provide important opportunities for dealing with resistance, for example, in diseases like chronic myelogenous leukemia. And the treatment of the relevant mutation with an allosteric inhibitor, which is already available, asciminib, bound to the myristoyl pocket at the C lobe of the kinase, can overcome drug resistance. So this is a good signal that these things can have practical applications, as shown by the image on the right-hand side. Next slide. I want to also give other examples of the potential for allosteric drugs in medical oncology. So in effect, trametinib, whose trade name is Mechanism, is a highly selective reversible allosteric inhibitor of MEK1 and MEK2 activity. I want to stress that it is in an ATP non-competitive inhibitor that binds MER adjacent to the ATP binding site. Of very great significance, it has been approved by the FDA for the combination treatment of unresectable metastatic melanoma with BRAF mutations in a very clinical applicable sense. I think it's important to remind everybody that once the Food and Drug Administration or EMA or other regulatory bodies approve an agent, that is a remarkable demarcation for both clinical and basic research. Allosteric therapy may have implications in the sarcomology2, so-called SH2 domain containing protein tyrosine phosphatase 2, the so-called SHP2 target that's encoded by PTN11. You've already heard a little bit about this already. The latter is a critical allosteric phosphatase for many signaling pathways. SHP2 is a non-receptor protein tyrosine phosphatase encoded by the PTN11 gene, and it is critical in cell growth and differentiation via MACD signaling pathway. SHP2 also plays an important role in programmed cell death pathway, effectively what is called the PDL1 pathway. Mutations in the gene that encodes for SHP2 have been linked to a variety of cancers, while SHP2 also enables activity in the cancer-associated RAS-MAP signaling pathway. I want to emphasize briefly the PDL1 part of this story, partially because that target is emerging as one of the most important targets, not only in oncology, but in all of pharmacology and pharmaceutical development. By the year 2025, it is estimated that Keytruda, which belongs to this class of therapy, will have revenue of 25 billion with a B in the year 2025. So we're talking about major public health and commercial implications linked to some of the processes that we're talking about today. Allosterically controlling the ubiquitin proteasome system has important implications in cancer therapy on a wide scale. And there are enormous, strong implications for allosteric interventions targeting tumor growth directly and targeting the tumor microenvironment. And this is extremely important because we now are realizing that not only do we have to worry about the tumor per se, but the tumor microenvironment, which can have enormous controlling features as to whether a patient will respond or not respond to therapy. Just as an example, the action of the collagen binding receptor tyrosine kinase discoidin, the discoidin domain receptor 2, DDR2, in both tumor and tumor stroma cells has been established as a critical feature for breast cancer metastasis. So these are important features of future research. Next slide, please. I want to end by talking about the future opportunities for Gain Therapeutics. I'm extraordinarily enthusiastic about Gain Therapeutics based on my interactions with the brilliant scientific staff at many levels. And I want to just indicate that proteome scale analysis, which we've discussed very briefly, of protein allosteric regulation perturbed by somatic mutations in at least 7,000 cancer genomes have already been reported in the literature, and I expect that list to keep going up. And this provides significant opportunities to utilize the CTX platform that we've heard about and related proprietary technologies at Gain Therapeutics. These technologies at Gain Therapeutics have already identified an allosteric target for a critically important oncogene. And again, I want to emphasize that there's great enthusiasm, not only within the pharmaceutical world, but in the various governmental bodies such as the National Institutes of Health and related agencies to pursue the goal of diminishing cancer mortality by at least 50%. And again, I emphasize that I think Gain will have an enormous role to play in this particularly important goal. And I want to thank everyone for their attention. Thank you so much.
Thank you so much, Dr. Broder, for that